Today we are going to study about cholesterol biosynthesis. Cholesterol biosynthesis in vertebrates takes place in liver. All growing animal tissues need cholesterol for synthesis of membranes, and some organs require cholesterol like adrenal gland and gonads, where they use cholesterol as precursors for hormone synthesis, steroid hormone synthesis. Cholesterol is also a precursor of vitamin D. Synthesis of cholesterol takes place in 4 stages. In the first stage, condensation of 3 acetate units takes place to form a 6-carbon intermediate called as mevalonate. In the second stage, mevalonate is converted to activated isoprene units, that is delta 3 isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. Then polymerization of 6 5-carbon isoprene units to form 30 carbon atoms choline takes place in the third stage. And in the last stage, cyclization of squalene to form 4-ring structure of steroid nucleus takes place, with further changes like oxidation, removal, or migration of methyl groups to produce cholesterol. In the first stage, formation of mevalonate takes place. Two acetyl-CoA molecules condense together in presence of thiolase enzyme. Coenzyme A is released and aceto-acetyl-CoA is formed. Aceto-acetyl-CoA reacts with another molecule of acetyl-CoA in presence of enzyme HMG-CoA synthase. Coenzyme A is released and beta-hydroxy beta-methyl glutaryl CoA is formed. This HMG-CoA is acted upon by HMG-CoA reductase enzyme. It uses 2-NADPH plus 2H plus as reducing equivalents, and which are converted into 2-NADP plus, and coenzyme A is released, and in this reaction mevalonate is formed. In the second stage, 2 isoprene units are formed from mevalonate. In the first reaction, mevalonate is activated by mevalonate 5-phosphotransferase enzyme. ATP acts as a phosphate group donor and is converted into ADP, and 5-phosphomevalonate is formed. 5-phosphomevalonate is acted upon by phosphomevalonate kinase. ATP is again the phosphate group donor, which is converted into ATP, and 5-pyrophosphomevalonate is formed. 5-pyrophosphomevalonate is acted upon by pyrophosphomevalonate decarboxylase. ATP transfers its phosphate group and is converted into ADP. An activated intermediate is formed, a transient intermediate is formed, which is called as 3-phosphor-5-pyrophosphomevalonate. Again, this intermediate is acted upon by pyrophosphomevalonate decarboxylase enzyme. Carbon dioxide is released and pi group is given off to form the first isoprene unit, that is delta 3 isopentenyl pyrophosphate. This delta 3 isopentenyl pyrophosphate is acted upon by isopentenyl pyrophosphate isomerase enzyme, and the second isoprene unit is formed, that is dimethyl allyl pyrophosphate. In the third stage, condensation of 6 isoprene units takes place to form squalene. First, dimethyl allyl pyrophosphate reacts with delta 3 isopentenyl pyrophosphate in presence of enzyme prenyl transferase. PPI is released and the condensation takes place in head to tail fashion, that is one isoprene unit's head condenses with the second isoprene unit's tail to form geranyl pyrophosphate. This geranyl pyrophosphate condenses with another molecule of delta 3 isopentenyl pyrophosphate in presence of enzyme prenyl transferase. PPI is released and again the condensation is in head to tail fashion, and the product farnesyl pyrophosphate is formed. Farnesyl pyrophosphate condenses with another molecule of farnesyl pyrophosphate. NADPH plus H plus provides the reducing equivalent. 2 PPI are given off and the reaction is catalyzed by squalene synthase, and the product of this reaction is squalene. In the fourth and the final stage, squalene is converted into steroid nucleus, and then steroid nucleus is converted by many reactions into cholesterol. First, choline is acted upon by choline monooxygenase. Here, a molecule of oxygen enters, and NADPH plus H plus also provides a reducing equivalent. These one oxygen atom reacts with two hydrogen atoms given by NADPH plus H plus to form a water molecule. Another oxygen atom adds to choline to form choline 2,3 epoxide. This choline 2,3 choline monooxygenase is a mixed function oxygenase. Now choline 2,3 epoxide by many reactions in plants gets converted into stigmasterol. In animals, by many reactions it is converted into lanosterol, and then lanosterol by many reactions is converted into cholesterol. In fungi, by many reactions choline 2,3 epoxide is converted into ergosterol. Regulation of cholesterol synthesis is regulated. In the mammals, intracellular cholesterol concentration as well as hormonal action like glucagon and insulin, they control the cholesterol synthesis. The rate limiting step and major site of cholesterol synthesis is that reaction which is catalyzed by HMG-CoA reductase enzyme, which converts beta-hydroxy beta-methyl glutarate CoA into mevalonate. This enzyme exists in two forms. One is phosphorylated form, which is inactive. The other is dephosphorylated form, which is active. Insulin promotes dephosphorylation. It stimulates dephosphorylation of HMG-CoA reductase enzyme and thus activates this enzyme, thus increasing cholesterol synthesis. While glucagon stimulates phosphorylation of this enzyme, due to which the enzyme becomes inactive and thus rate of cholesterol synthesis decreases. Another unidentified X represents unidentified metabolite of cholesterol that inhibits or decreases HMG-CoA reductase enzyme's activity because it promotes proteolysis of HMG-CoA reductase. When the cholesterol concentration, intracellular cholesterol concentration increases, it activates enzyme acyl-CoA cholesterol acyltransferase, which esterifies cholesterol. So more of cholesterol esters are formed. And another thing is this X also inhibits uptake of LDL cholesterol by cells. So in this way, cholesterol concentration is regulated in the cell. HMG-CoA exists in two forms. One is phosphorylated and the other is dephosphorylated form. Phosphorylated form is inactive and dephosphorylated form is active. Glucagon stimulates phosphorylation, hence inactivates enzyme, while insulin promotes dephosphorylation, thus activating the enzyme. High intracellular concentration of cholesterol activate acyl-CoA cholesterol acyltransferase, which increases esterification of cholesterol for storage purposes. High cellular cholesterol levels diminish transcription of the gene that encodes LDL receptor, reducing production of the receptor and thus uptake of cholesterol from blood. In this way, levels of cholesterol in cell are regulated.